Today, we are diving into some really fascinating new research on an illness that is notoriously tough to handle: Lyme disease. And it all revolves around one single powerful question.
This, right here, this is the whole game. For the thousands upon thousands of people who suffer from symptoms that just won't go away, what if the answer isn't a new drug, but a way to actually retrain our body's own defense system? It's an incredible idea, and we're going to unpack it right now.
Okay, so let's get into it. Before we can even start talking about a potential solution, we really need to understand the problem itself. And with Lyme, it's complicated. So, the basics, right? Lyme disease comes from a bacteria called Borrelia burgdorferi, and you get it from an infected tick. And this is not a rare thing. We're talking over 300,000 new cases every year in the US alone. Now, for a lot of people, if you catch it early, a round of antibiotics can knock it out. But, and this is a huge but, for about 10 to 20% of people, the fight isn't over. Debilitating symptoms like crushing fatigue, joint pain, and that awful brain fog can stick around long after the treatment ends. This is what's known as post-treatment Lyme disease syndrome, or PTLDS. And that leads us to the million-dollar question that has baffled doctors and patients for decades. If the antibiotics wiped out the bacteria, why are people still so incredibly sick?
Well, this quote points to a major shift in thinking. More and more research is suggesting that the real culprit might not be a stubborn, hidden infection. Instead, the theory is that the immune system itself has gone haywire. It's stuck in this overdrive inflammatory state, even though the original threat is long gone.
So if the problem is an out-of-control immune system, then what's the solution? Well, what if you could retrain it? Send it back to school, so to speak. This is where the science gets really, really cool.
The key word here is immunomodulator. Now, that sounds complicated, but think of it this way. It's not just a blunt instrument that either boosts or suppresses your immune system. It's more like a skilled conductor leading an orchestra, telling one section to play louder while telling another to quiet down, creating a balanced, perfect response.
And this brings us to a very special kind of immunomodulator: beta-glucans. These are basically natural fibers. You find them in the cell walls of things you know, like baker's yeast, shiitake mushrooms, oats. And you can almost think of them as a training gym for your immune cells, getting them in shape to be smarter, more efficient, and more balanced. So how in the world does that work? It's actually pretty elegant. When you consume a beta-glucan, it travels down into your gut. There, your immune cells see its unique structure and recognize it as something foreign. Now, this doesn't trigger a massive all-out war. Instead, it just sort of primes the system. It puts your immune cells on a state of alert, getting them ready to respond much more effectively when a real threat comes along. Okay, so that's the theory. Sounds pretty good, but what happens when you actually test it against Lyme? Let's look at the evidence, starting with a really key animal study. So here's the setup. Researchers took a specific beta-glucan that comes from yeast, and they gave it to mice. Then they infected those mice with the Lyme bacteria to see if this immune priming actually made a difference in how the disease progressed. And the results were, well, pretty incredible. A classic sign of Lyme arthritis in mice is severe ankle swelling. Just look at this chart. The control group on the left, lots of swelling. But the mice that got the beta-glucan, way, way less. That's a direct physical measurement showing the disease was less severe. And it wasn't just the swelling. This table shows the scores for how bad the arthritis and carditis, that's inflammation of the heart, actually got. And on both counts, the glucan group scored significantly lower, which means they were healthier. So the big takeaway here is that at least in this animal model, yeast-derived beta-glucan could directly reduce the physical damage of Lyme disease. All right, those are some powerful results in mice. But the question everyone always asks is, that's great, but does it work in people? So let's turn to a human pilot study. But I need you to pay really close attention here, because there are some key differences. This slide is so important to understanding where the research is right now. The mouse study we just saw used a beta-glucan from yeast. The human study used a related but different compound called AHCC. And AHCC is an alpha-glucan from shiitake mushrooms. They both might work on the immune system, but they are not the same thing. It's a critical distinction. And here's another piece of crucial context. This was a very small preliminary study, just 11 patients. It was also open label, which means there wasn't a placebo group for comparison. Everybody got the AHCC. This kind of study is really just meant to see if there is a signal,
A little hint that it might be worth investigating more. So, did they find a signal? You bet they did. First, they looked at a general marker of inflammation in the body. It's called ESR. Think of it as a body-wide inflammationometer. And look what happened. After 8 weeks, the patient's average levels dropped significantly. That's a very clear sign that inflammation was cooling down.
They also looked at more specific inflammation messengers. And this one, interleukin-8, is a big deal. Why? Because we know that IL-8 levels are often cranked way up in Lyme patients. To see the specific marker go down so much is a really promising sign.
But okay, lab markers are one thing. How did the patients actually feel? Well, this might just be the most amazing finding of all. Every single one of the 11 patients, that's 100%, reported that their worst symptoms, the joint pain, the muscle issues, the flu-like feeling, had either completely or at least partially resolved after those 8 weeks. Okay, let's take a breath. We've got a really strong animal study using a yeast beta-glucan and a very suggestive small human study with a related mushroom alpha-glucan. So, what does it all mean? Let's put the pieces together. So here's the bottom line. This is incredibly promising, but it's not yet proven. The animal data with yeast-derived beta-glucan is solid. It shows a direct reduction in physical symptoms. The human data with AHCC is suggestive. It points toward lower inflammation and people feeling better. The path forward is crystal clear. We need bigger, placebo-controlled human trials to really confirm all this. And that leaves us with this final, really provocative idea to think about. This research, as early as it is, could be pointing us in a radical new direction. For Lyme, and maybe for a lot of other complex post-infectious illnesses where the immune system seems to have gone rogue, could the future of medicine not be another pill to kill a bug, but a tool to train and rebalance our own internal army? It's a powerful thought, and science is just starting to scratch the surface.